Hello everyone, welcome back to Prep Pro Papers. So we are now at part 4 of GS 2023 TIFR JGEEBILS paper. Let's start with the biology section. The first question says, in a human family, the father and the mother are carriers for an autosomal recessive disease. One of their son is phenotypically normal. What is the probability that this son is also a carrier? Okay, so let's consider that if the father and the mother are the carriers for an autosomal recessive disease, so they are not disease. That means the condition is heterozygous. So the possible combinations can be capital A, capital A, capital A, small a, capital A, small a, and small a in the ratio of these genotypical progenies which we got will be 1:2:1. So in this case we have to find out if the son is also the carrier. So as the disease is autosomal recessive, the recessive genes are found in these two places. So the probability of the son to be the carrier is 2 + 1 is the total condition, and the son to be just the carrier, this is the diseased phenotype, is 2/3. Option number 4. Next question says, which of the following techniques can be used to identify post-translational modifications such as phosphorylation or methylation of a protein and the residue where it is modified? So let's see by the option. The first option says 2D electrophoresis. So the 2D electrophoresis will only help for the knowing the separate proteins. Only helps to separate the proteins. Size exclusion chromatography and even the UV visible spectroscopy, these are not pinpointing specific post-translational modifications. Now for the mass spectroscopy, these are this give us the detailed information about the molecular weight and the structure of peptides and protein. So the most relevant option will be option number 3, mass spectroscopy. Let's go to the next question. Amino acids have different chemical properties that determine whether they are found on the surface of globular proteins. Which of the following series of amino acids is ordered according to how likely it will be found on the surface of a protein? So if we are talking about whether the protein are found on the surface of the globular proteins, these property will depend on the polarity and hydrophilicity of the proteins. So let's check upon the options. Arginine is basic and polar, so it's likely to be found on the surface. Leucine is non-polar, so it's not likely to found on the surface. Aspartic acid is acidic and polar, so likely to be found on the surface. And phenylalanine is non-polar, so not likely to be found on the surface. So leucine and phenylalanine should be less likely than aspartic acid. So this order is not correct. Let's check for the second option. Threonine is polar and uncharged, so it's likely to be on the surface. Arginine is basic, polar, so likely to be on the surface. Phenylalanine is non-polar, so not likely to be on the surface. Asparagine are polar and uncharged, so likely to be on the surface. So except for phenylalanine being misplaced among the more polar residues, so this option is also not correct. Let's check for the another option. Arginine is again basic, polar, so likely to be on the surface. Phenylalanine non-polar, so not likely to be on the surface. Threonine is polar, uncharged, so likely to be surface. Glycine is non-polar and is very small, so phenylalanine and glycine placement in this order is not correct, so again this order is also wrong. The last option says aspartic acid is acidic, polar, so likely to be on the surface. Threonine is polar and uncharged, so likely to be on the surface. Leucine is a non-polar, so not likely to be on the surface. And phenylalanine is also not polar, so not likely to be on the surface. So the correct option is option number 4. Let's check the another question. You synthesize a short peptide named this, where X is in any amino acid with a charged side chain. How many possible peptides can be synthesized? So we have 3 places where X has been placed. We know there are 5 amino acids with charged chains. These are aspartic acid, which is ASP is a short form and D as an abbreviation form. Glutamic acid, is written as E. Lysine as K. Arginine as R, this is K, and histidine as H. So if there are 5 charged side chains of an amino acid and the possible places are 1, 2 and 3, so the total combination will be 125. Option number 3. Let's go to another question. Human DNA contains 18% of C on a molar basis. What is the mole percentage of A, G and T respectively? So we know that A bonds to T, G bonds to C, A and G are purines, T and C are pyrimidines. So we know that the percentage of the purines is equal to the percentage of the pyrimidines. So if C is 18%, so G will be 18%. So this will be 18. In total, the concentration will be, as we know, A plus C will A plus T will be equal to G plus C also. Sorry, I made it wrong. A plus G will be equal to C plus T. So this will be total of 16%, 36%. From 36%, the A and T will be 100 minus 36, that will be 64. So if 64 is the total concentration of A plus T, so each will be 64 by 2, that is 32. So option number 1 is the correct option. Next question is just easy one, which you can just eliminate by the options. Which of the following statement explain why net primary productivity is higher near the tropics? More predators in the tropics kill herbivores, increasing plant growth in the tropics. No. Increased species diversity results in increased plant growth. No. Availability of newly weathered inorganic nutrients. No. Water and warm temperatures in the tropics promote the photosynthesis. Yes, the water and the warm temperatures are the specificity to the tropics. So this is the correct option. Option number 4. The next question says, it's a lengthy one. Let's read it. The stages of the cell cycle for a cultured mammalian cell line required the following periods of the time. G1 is equal to 8 hour, S is equal to 5 hour, G2 is equal to 1 hour, M is equal to 1 hour. An asynchronous culture of these cells is exposed to radioactive thymidine for 5 minutes and then allowed to continue to grow in non-radioactive medium. The figure below shows the results. If the length of G2 were increased by 1 hour, which of the following statements would be true regarding the slope of the line from the first appearance of labeled mitotic cells until all cells are labeled? Okay. So let's take for two conditions. First, originally G1 is for 8 hours, S is for 5 hours, G2 is for 1 hour and M is for 1 hour. So total it is of 15 hours. Now when G2 phase is increased by 1 hour, G1 is again similar 8 hours, S is 5 hour, G2 is 2 hour and M is 1 hour. The total time is here 16 hours. Now listen up carefully. The key point is the slope of the line from the first appearance of labeled mitotic cells until all cells are labeled. This period is determined by the time it takes for cells that have incorporated the radioactive label during the S phase to progress through G2 and M phase until they are detected in mitosis. Because the radioactive labeled cells must still pass through the same stages of G2 and M, about 1 hour longer in G2, the rate at which labeled cells appear in the mitosis depends upon the intrinsic labeling period and consistent division rates, meaning the relative intervals and appearance rates remain proportionally unchanged. So the correct option will be option number 3. It will remain the same. Next is again an easy question. X-linked recessive disorders are most commonly exacerbated because females, because they have 2 chromosomes and are therefore more likely to inherit one with a disease. No, that's a half case. Males, because they have only 1 X chromosome. Yes, because a single recessive gene on the X will cause the disorder, because there is no corresponding gene on the Y chromosome to counteract it. So the correct option is option number 2. Next is again an easy question. Which of the following statement is correct given this phylogeny for 4 species? A has gone extinct. No. E is more closely related to C and D. No. D evolved from C. No. D and C evolved from the common ancestor. B, C and D and share a common ancestor. B, C and D. Yes, this is the common ancestor they are sharing. So the correct option is option number 4. A trihybrid test cross order of loci unknown produces progeny classes as follows. The gene order is. Okay, so this question might seem a smaller one, but yes, it is again an easy one, but let's see how we solve this type of questions. So the first thing we have to find out is the parental type and the double crossover types. For the parental type, the most frequent progeny are considered to be parental type. So here is this and this as parental type. Let's write it down. 35A small b capital C and 37 small a capital B small c. For the double crossover, it is the least frequent progeny. In this case, this is this one. Let's write it down. It will be 1 capital A small b small c and small a capital B capital C. So from if we check from parental type capital A B and capital A small b capital C to double crossover type capital A B small c, so A and B stays the same here and capital C changes to small c. This indicates that the middle gene should be changing between the parental and the double crossover type. Now we check from capital A, small a, capital B, small c to small a, capital B, capital C. The A and B positions are same and capital C again changes to small c again changes to capital C. So in both the double crossovers, the C gene is the one that changes between the parental and the double crossover, indicating that the C is the middle gene. So let's check upon the option where C is the middle. The correct option is option number 3. Let's check for the next question. A newly discovered extraterrestrial organism was found to contain proteins that are made up of 75 different amino acids. Assuming that one codon codes for a unique amino acid, which of the following is possible correct about its DNA codons? Okay, again a simple question. We just need to check whether the number of amino acids coming will be greater or lesser than the 75 desired amino acids. So the first option says the DNA could be composed of 4 bases and the organism has a triplet codon, which is 64 amino acid, which is lesser than the desired amino acids count. The second option says the DNA could be composed of 3 bases and the organism has a triplet codon. The DNA could be composed of 3 bases and the organism has a quadrat codon. So it's 3 to the power 4, 81, greater than 75. So our desired number of amino acids can be covered in this. But let's check for the next question option also. The DNA should be composed of 2 bases and the organism has a quadrant codon. It is 16, again lesser than 75. So the correct option is option number 3. You unearth a fossilized skull. Your initial investigation suggests a reptile, likely a dinosaur, that had strong jaw muscles. What feature led to you that conclusion? The presence of sagittal crest? Yes. So listen carefully. Because a sagittal crest is a bony ridge on the top of the skull to which jaw muscles get attached. its presence indicate that the animal had strong jaw muscle, which is often associated with the powerful biting and chewing capabilities. This feature is commonly found in dinosaurs and other animals that require strong jaw muscles for their feeding habits. So the correct option is option number 1. The mutation rates due to replication error in strains E and B of E. coli are 10 to the power minus 8 and 10 to the power minus 9 per genome per generation respectively. Starting from the 10 cells of each generation, the bacteria will culture for varying amounts of time as given below. Both strains divide once in 20 minutes. In which of the cases would both cultures have at least one mutation of bacteria? Okay, so let's go option by option. This is a lengthier question. I will copy this section. Let's solve for here. First, we need to know that this compare for strain A and B. Both the strains divide every 20 minutes. And they both started with 10 cells each. The mutation rates for A it is 10 to the power minus 8 and for B is 10 to the power minus 9 cells per generations. So we need to find the time at which the number of cells ensure there is at least one mutant bacterium. So the doublings in 20 minutes can be written as time required upon 20 minutes, and the total cells after time t will be 10 into 2 की power t upon 20. So let's calculate. Okay, so this is for A, which we copied. So for 520 minutes, the doublings will be, I am writing doublings at D as 520 upon 20, which will be 26. So total cells will be 10 into 2 की power 26, which will be 671,088,640. There is a calculator given in TIFR exam, so you can use in this scenario. Otherwise, I'll say not to use a calculator because it consumes lots of time. So the mutants will be 671,088,640 into 10 to the power minus 8, which will be coming from 6.71, which are enough. For 120 minutes, the doubling time will be 560 upon 5 out 560, it's 120, which will be 120 upon 20, which is equal to 6. Total cells will be 10 into 2 की power 6, which is 640. So the number of mutants will be 640 into 10 to the power minus 8, which will come to be around 0.0064, which are very less, not enough. For 560 minutes, the doubling times will be 560 upon 20, which will be equal to 28. Total cells will be 10 into 2 की power 28, which is 2684354 and the number of mutants will be 2684354 into 10 to the power minus 8, which is 26.84, which are enough. For 500, the doublings will be 500 divided by 20, which is 25. Total cells will be 10 into 2 की power 25, which will be 335,544,320. So the number of mutants will be 335,544,320 into 10 to the power minus 8, which is 3.36. It's again enough number of mutants per generations. Now let's check for strain B. I'll again copy this. Okay, so for 400 minutes one, the doubling will be 400 by 20, which will be equal to 20. Total cells will be 10 into 2 की power 20, that will be 10, 4, 8, 5, 7, 6, 0, and the mutants will be 10, 1, 0, 4, 8, 5, 760 into 10 power minus 9, that is 0.0105, which are not enough. For 240 minutes, the doubling time will be 360 divided by 20, which will be coming to be 18. Total cells will be 10 into 2 की power 80, will be 2621440. The number of mutants will be 2621440 into 10 to the power minus 9, which will be 0.0026. Again, not enough. For 360 minutes, the doubling time will be 360 upon 20. Total cells will be 10 into 2 की power 18, 2621440. The number of mutants will be 2621440 into 10 to the power minus 9, which will be 0.0026. Again, not enough. For 560, it will be double time will be 560 divided by 20, 28. Total cells will be 10 into 2 की power 28, which will be coming to 2684354,560. The mutants will be 2684354,560 into 10 to the power minus 9, which will be 2.68, which are enough. So comparing both from the strain A to the strain B, the correct option for this is option number 4. I know it is the lengthiest question, but I think this paper of 2023 contains a lot of lengthy question, and yes, conceptual ones were also there. Compared to 2024, I'll say it's more difficult, but still the entire paper will be in moderate level. Let's check upon the last question. Scientists measured the resting metabolic rate RMR of males and females from 12 different insects species. Given the results shown In the figure, which statement is correct? Circles with a cross indicate the species that eat plants. Okay, this type of question you just have to eliminate through the options. In each species, males and females have similar RMR. No, there is no two corresponding lines which says that they have sharing the similar RMR. So this option gets eliminated. Plant eating insects have higher RMR than other insects. These are the plant eating insects one, but they are showing lower RMR level compared to the other insects. So again, this option is wrong. Male and female RMR are not correlated. No, they are correlated because they are more likely to be at the particular trend line. So again, this option is eliminated. Males generally have higher RMR than females. Okay, so we are seeing that the males are showing higher RMR value compared to the female ones. So yes, this option is correct. So hope you are liking our series. The exam is coming forward. Please check upon our previous year question papers of 2024 GS and 2023 GS is now completed. I hope you do well in your exams and you're liking Prep Pro papers initiative. Just a new announcement that we are coming with the Python and machine learning, which is again a very important aspect in nowadays in every field. Please do check upon that. That will be uploaded soon in this week. We will likely to be complete more of the TIFR JGEEBILS within this week, so that it will ease your paper on 8th of the December 2024. Thank you and hope you like it. Please subscribe the channel and hit the bell icon to know more.